So, memory, it's about time. But is it really about time? Just firstly, there's this very long argument about interference versus decay, right? I mean, have the decay people won? Because I think the interference people are still, still fighting. They're still fighting, right? So is it really about time? And what about rehearsal? Think about working memory, like good old Baddeley style working memory. You can get amnesics to rehearse stuff for an hour in the dark or whatever, and they'll do it, right? So it's working memory is kind of timeless, right? So yeah, just saying it's not about time. But actually, today I'm going to say that it is about time. And before I say that, I just want to say, let's think about some ways it might not be about time, right? So imagine a view of memory where, we do things like perception and decision at these different moments. And then memory is this thing, it's like a crane that reaches back and it gets stuff from the past and it brings us back to the present, right? And I think that sometimes we think about memory like this. And then sometimes we think about memory like this, where processes themselves are kind of extended in time and overlapping. And it's not clear to me that when we say memory in these two cases, they were talking about the same kind of thing. Maybe we can bring this back in the discussion. What I'm going to say today is that it really is about time. And I'm going to present some evidence for timescale hierarchy in the human brain. I'm going to start by saying that information flow is organized hierarchically, which is a very familiar idea. Then I'll say that the higher order regions are actually integrating information over longer time windows. And then I'll say that it really is something about time, because there's something about the relationship to the dynamics in these areas and their anatomy and so forth. So that's the picture of what I'm going to say today. And I'll start with this hierarchy idea. So imagine that you just want to know, you want to just count steps from the outside world into the brain. The outside world is my eye, and then I count steps, thalamus, V1, etc. And I count steps, right? And I want to ask, where's the outside and where's the inside? What's the most outer part? What's the most inner part? I can take data like this, or I could take functional connectivity data, and I can derive maps that look kind of like this. These areas in red here are one way of mapping the furthest points from the sensory periphery. And you'll notice that these areas, the areas in red here, we can call them the interior world. We call the blue areas the exterior world in the cerebral cortex. They look a lot like areas in the default mode network. And we can ask what kind of information is processed in these most interior parts of our brains. We did an experiment many years ago now, where we played people movie clips, and some of the movie clips were intact and some of them were scrambled in time. So I'm going to give you an example of what the intact clip looks like. This is a very high quality movie from 1975 called Dog Day Afternoon. I highly recommend it. Okay, this is the clip.
Nobody move! Get over there! Okay. All right, get away from those alarms. Good! Now get in the center. He moves, take his head off. Put the gun on him. Get out of the center.
Sonny? I can't do it, Sonny.
What?
I'm not gonna make it, Sonny.
What are you talking about? Put it on him.
I can't do it, Sonny.
Oh, fuck me. Sal. Sal, what? Where are you?
So that's the intact clip. Here's the scrambled version of the clip.
Okay, take care of it. Gotcha. Gotcha. All right? Okay. Cheer up. God! The keys.
So you notice that when you watch the first clip, Al Pacino walks into the bank and he has this kind of relationship with the tellers that it builds up over time. You see the scene from different angles. You discern the relationship between him and his partner. You're getting all the same visual information, all the same auditory information in these two clips, but one is a continuous kind of narrative and one is not. You can map neural responses to the various clips here. You can measure which parts of the brain are responsive to these movie clips. This is a map of responsive regions. The areas in blue are areas that are only entrained by the intact movie and not by the scrambled movies. And these areas are the same areas. It is the interior world, okay? It's the interior world. And so we could say something like, narratives reach deep inside of us, okay? And Meshulam in 1998 had this beautiful review paper where he talked about kind of this kind of big picture view of cognitive and neural architecture. And he talked about the external world and the internal milieu. So, V1 here is the beginning of this visual pathway deeper inside us. And the A1 is the beginning of this blue auditory pathway deeper inside of us. And right here, very deep inside of us, is our default mode network. And narratives somehow are in engaging activity in these areas. They're entraining the entire hierarchy all the way through to the DMN. Now, so, when we ask what's the function of default mode network regions, I'm sure everybody here is familiar with this. There are many different names for this network. It's called the default mode network, the mentalizing network, the narrative network, the emotion network, the memory network. One way of summarizing what it's involved in is representations of who, what, why, and where, representations of yourself into other scenarios. It represents situations. Okay, so one way of describing what I've just said is that there's a hierarchy of abstraction from the outside world inside of us, right? So the outside world is close to kind of sensory features, and the inner world is close to representations of the self and situations. And that's roughly right. So information flows organized hierarchically between periphery and interior. And now I'm going to say that there's another way of thinking about this, right? And I'm going to say there's a timescale hierarchy in the human brain. And I'm going to show you an experiment that was led by a grad student in my lab who's defending her thesis in a few days and going to become a AI researcher in a wonderful AI lab. So, okay. So the question now is, I've said that there's a timescale hierarchy in the human brain. How can we measure the timescales in each particular brain region? What we're going to do is we can say, here's an intact stimulus, a little bit of speech, right? I'm 5,000 feet above Albany on a beautiful day, beautiful blue sky. And I can measure the neural response at this moment. I can also measure the response to the same words, beautiful blue sky, but preceded by some different context, right? And you can say that if the response in the two cases is the same, then this is a particular brain region you're measuring from that's just not sensitive to the events from before. So its time scale has to be shorter than this black box, roughly speaking. But if you measure the response and it is different, it means that the time scale has to be a little bit bigger than that black box, at least, right? That's the logic. Now, we came up with something a little bit, kind of a way to map timescales in the cerebral cortex that uses this kind of reasoning. So what we're going to do is we're going to have two groups of participants, and they're going to be hearing some sentences in a narrative that are shared, but they're going to be preceded by differing contexts. So you're going to have the the little gray tokens over here and the little black tokens. At some moments, they'll be hearing exactly the same materials, right? And in some cases, they'll be hearing totally different materials. And what we're going to ask is, after they start to hear exactly the same input, how long will it take their neural dynamics to converge? So for how long will it take for a given brain region for the gray tokens and the black tokens to kind of converge in their neural responses, right? So you're getting exactly the same input, but the contexts are different. We predict that if an area has a short time scale, the responses should converge quite shortly after the onset of the shared input. But if you have a long time scale, in some sense, your kind of processing window is going to be hanging on to this context from the past, right? And so it's going to take longer for you to have enough shared context for your neural responses to look the same. Does that make sense? So, and there's also another part of the experiment that I won't have time to talk about today, where people separate their responses. So we're going to be making our dependent variable is going to be inter-subject pattern correlation. So we're going to be taking a particular brain region. We're going to be looking across these two groups, and within the two groups. We'll be looking within, let's just say segment 5 of our story here. And we're going to be measuring, on the x-axis here is time from the onset of the sentence. On the y-axis here is voxels. So for one person, I have a measurement of their response to this text, the speech at the beginning of number 5. And in the other person, I have their response. And our measurement will be how similar are these vectors. So how similar is the top red rectangle to the bottom red rectangle, etc. So at each time point, we can measure that. That's how we're going to measure the alignment of the two groups across contexts. Now, we can also do this within groups. So for two people who are hearing exactly the same stimulus the entire time with the same context, we can also measure how similar their responses are as a function of time. So I'll give you an example of what this looks like. In auditory cortex, right, the response within the intact group is very stable. So on the x-axis here is time from the onset of a sentence. On the y-axis is how similar are individuals to one another, and they're similar, and it's very stable over the sentence. In the group of people hearing the scrambled version of the story, they're hearing the same sentences in a scrambled order, they also have a reliable response from the beginning of the sentence all the way to the end. Then in the cross-group comparison, our key comparison, the responses are different at the beginning, but then it ramps up. The reason they're different at the beginning is because of hemodynamics. In one group, they're hearing the same input now, but the hemodynamic response that we're measuring is from the previous sentence, which is different in these two groups, right? So that's why it starts, the correlation starts at 0, but as the hemodynamic effect wanes away, the cross-group similarity is as high as the within-group similarity. Now I'll show you an example from temporoparietal junction. The responses are stable in the intact group and in the scrambled group. But when we look at the cross-group comparison, we find that these two groups are producing totally distinct responses for 7 seconds into this sentence. So just to be clear here, we have two groups of subjects. Each group is producing a stable response to the stimulus. We can, decode what sentence it is and what moment it is, right? But the two responses are just totally different to the same input. Okay, so for every brain region, we can make maps like this green curve, right? We can make a curve like this for every region that responds to the stimulus. So for example, here are RI by RI maps. These are ones that ramp pretty quickly. These are some ones that ramp a bit slower. These are ones that ramp slower still. And these are ones that ramp really slowly. It takes them on the order of 10 seconds before the responses align to exactly the same input. And we characterize this ramping using a logistic fit. So we can characterize, we call this the alignment time, which is the time it takes for this ramp to get to half of its maximum. And if we map this on the cortical surface, we can color cortical areas by when the alignment takes place. The areas in dark maroon are the areas that ramp quickest. They're the sensory areas. And as you can see, as you move back from more sensory areas towards areas like the TPJ, you start to find areas that don't align across individuals until 8 to 10 seconds after the sentence onset. Okay, so the claim here is that really these higher order areas must have some kind of extremely context dependent response that's spanning quite a long period of time, many seconds, in order to exhibit a response like this. Because to be clear, both groups are responding reliably to the stimulus. It's the same stimulus, but the responses are different. Okay. And the data and the code for this are available. The data on OpenNeuro and the code is on our lab GitHub. This finding is not specific to this study. You can get similar kinds of effects with auditory stories in other analyses. You can do this, it doesn't require language. You can do it with silent movies. It's not specific to fMRI. We did it with intracranial recordings. My claim is that the timescales of information processing vary hierarchically across the cortex. And the idea is that as you move from outer to innermost parts of the cortical network, brain regions integrate information over longer windows. Okay. Now, let's ask about, is this really about time, right? And I'm going to say that I think it is, right? Oh, one other thing to say is that in the study that I just told you, there's a whole lot more. We show that the integration process is gated at event boundaries, and we build a computational model to account for it. So check it out. Okay, is this really about time, or is it just about preferred kinds of information, right? So that example with Al Pacino at the beginning, is it really about time, or is it just the fact that like the intact movie just has information in it that the other movie, the scrambled movie, doesn't have in it, right? It's really about time. So let's ask ourselves, where are brain dynamics intrinsically the slowest in the human brain? So one way to do this is with intracranial recordings, which we did, but another way to do it is with fMRI. You can just take bold signal and you can compute the power spectrum of resting state signals, and you can ask what those power spectra look like. So power spectra have this kind of 1/f structure in bold signal, and you can just cluster voxels according to those that have steeper 1/f structures and those that have flatter. The steeper ones have a larger proportion of slow dynamics. These are the slowest regions, the blue voxels, they're here. The fastest are those in red, they have the flattest spectra. And as you can see, there's this gradient that just maps from early sensory areas towards the TPJ in just the same way. And this is measurement of intrinsic dynamics. You can get the same map whether you measure eyes closed rest or if you have people listening to a narrative, and you get the same map if you if you average the responses across subjects to extract the stimulus locked response. Another way to look at this is many of you will be familiar with this paper from Chris Baldessano et al. You can take spatial patterns and ask how similar are they time point by time point, right? So this is a MVPA-ish kind of analysis. This is a time correlation map. You've seen many of these today. The time correlation map in V1 looks like this. So this is the diagonal, and these blocks, these little blocks are moments when the V1 patterns are pretty stable. And the patterns in posterior medial cortex look like this, right? So the blocks in posterior medial cortex are just way bigger than the blocks in V1. And you can kind of basically make a map of how big these blocks are on average, and it looks like this. And it's the same kind of story. You see this effect in single units, you see it in LFPs, you see it in intracranial recordings. The important thing to emphasize here is that it's not that there's one time scale in a given region, it's that there's a mixture of time scales in every region, but regions are biased to have a certain proportion of fast or slow dynamics. And also it's important to note that every region can rapidly shift its state. It's not the case that if you're a slow region, you can never shift quickly. I've got 2 more slides. Is that okay, Aaron? Okay. So you might ask, wait a second, but when we process information over time, sometimes things happen faster and sometimes they happen slower, right? So we did an experiment where we presented stories at double speed or at half speed, right? And if these timescales were really fixed and really rigid, then the kind of integration that would be happening in a 3-second window is going to be very different in the slow story and in the faster version of the story. And to make a long story short, these processing windows rescale. It's not that they're absolutely fixed time constants. Within about the range of about half speed to about double speed, these intrinsic time these processing time scales can stretch and compress. And there's another demonstration of this year with modeling from BU Her's lab with auditory musical stimuli. The final reason why it really is about time is that these timescale gradients correspond to anatomical connectivity, they correspond to functional connectivity gradients, they correspond to gradients of myelin density and gene expression, and they also, these intrinsic timescales that you can measure in fMRI are also predictive of clinical symptoms in autism, for example. They actually are prospectively predictive. So the claim, I really want to just make this claim that it really is about time. Is it really? Yes, it really is. Thank you very much.